Good afternoon. We are honored to invite you to this webinar. I'm Gina Liu, in charge of R&D department from Rocket Healthcare. Today, we are going to talk about hyperlyzed kidney treatment. In particular, today we'll explain the clinical application of R&D, single-cell R&D analysis in the last session. The webinar will begin with the opening ceremony of Professor James Yu. He works at Wake Forest University and he is a key opinion leader in the regenerative medicine. Next, we will introduce overall idea of our regenerative platform. Then, explain more detail in technology session. Please leave questions during the webinar. We'll answer them in Q&A session. I hope you have a great time.
Greetings. First, I would like to thank the seminar organizers and Rocket Healthcare for inviting me to kick off this online conference that focuses on kidney regeneration. It is an exciting topic to start the new year. Chronic kidney disease is a condition that often progresses to renal failure, and it is a leading cause of mortality and morbidity. Chronic kidney disease is characterized by severe loss of renal function, and approximately 11% of world's population progress to end-stage renal disease. End-stage renal failure is a devastating disease which involves multiple organs. Although dialysis can prolong survival of many of these patients, it is not a curative treatment. Only renal transplantation can restore a normal renal function. Unfortunately, renal transplantation is severely limited by donor kidney shortage. So patients are dying, even at this moment, waiting for transplantable kidneys. To overcome this challenge, many decades of extensive research have been performed to develop therapies that could restore normal kidney function. The kidney is a complex organ with many critical functions such as waste removal from the body and homeostasis maintenance. Recent advances in tissue engineering and regenerative medicine have offered many possibilities for developing therapies that could enhance and restore normal kidney function. An augmentation of either partial or total renal function using regenerative medicine approaches may be a feasible solution for these patients. Some of these strategies include cellular therapy using either stem cells or autologous cells, bioengineered implantable kidney tissues for partial or whole organ regeneration, and in situ tissue regeneration strategy that utilizes the body's biological resources to regenerate kidney tissues. The potential of these strategies is now possible due to the advances in various platform technologies, including cell technology, scaffold technology, bioreactor, and biofabrication technologies. At this conference, some of these technologies will be introduced and discuss how these technologies could be used to address the therapeutic challenges in chronic kidney disease. Specifically, this seminar will include the use of omental patch to augment kidney tissues and looks at safety and efficacy of this approach. The use of kidney-derived scaffolds is also presented, and methods to decellularize the scaffolds and repopulate the target cells will be discussed. Additionally, there will be a presentation on how single-cell RNA analysis could be applied for kidney regeneration. I hope you will enjoy the seminar. And again, I thank Rocket Healthcare for having me here today. Thank you.
Hello, my name is Jina Ryu. I'm in charge of kidney regeneration platform in Rocket Healthcare. Today, I'd like to talk about hyper-personalized kidney treatment, which is an AI-based 3D printing technology and single-cell analysis for chronic kidney disease. According to the World Health Organization report, in 2020, the global population aged 60 years and over is just over 1 billion people, and that number is 2.5 times greater than in 1980s. As life expectancy increases, humans can live over 100 years. But there's a lifespan in the organs. When you see this picture, skin has the shortest lifespan, from around 18. Resilient collagen and stretch elastin decline at about 1% per year. The longest brain also have age-related change by 70. Since conventional treatments have applied one drug to all patients, side effects were found in some patients. Moreover, a lot of time and money were spent on clinical trial. Therefore, Rocket begins to try personalized treatment using a 3D bioprinter with bioink derived from the patient's own tissue. Founded in 2012, Rocket Healthcare has developed a 3D bioprinter and used it to treat chronic disease such as diabetic foot ulcer, osteoarthritis, and chronic kidney disease. 3D bioprinter, AI application, medical device kit, and bioink are combined to offer a safe, effective, and high quality of solution for chronic disease. All of these technologies are patented and the treatment protocol are approved as non-ATMP by the EMA. In addition, 3D bioprinter and AI application and medical kits have been approved by FDA as medical devices. With the core technologies, we developed therapeutic patch platform for chronic disease. We applied our platform to diabetic foot ulcer, osteoarthritis, and chronic kidney disease. Since conventional monotherapy was not effective for chronic disease, Rocket's multitherapy using autologous tissue can be a solution to treatment chronic disease. Today, we are more focused on chronic kidney disease treatment. Before I move in CKD treatment, I will introduce briefly about DFU and OA. DFU caused by complications of diabetes, and it was not treated well with a conventional dressing treatment. However, Rocky's therapeutic patch made with patient own fat showed nearly 90% recovery rate. This clinical study was performed with Dr. Armstrong, who is an internationally recognized leader in the field of DFU. Next clinical application was osteoarthritis. From the preclinical study with Harvard Medical School, we found that cartilage was regenerated after patch treatment. After confirming the efficacy and safety from animal model, we had a clinical study in Egypt. The clinical study results are preparing for publication. The next clinical application is chronic kidney disease. It is a treatment to attach therapeutic patch made with 3D bioprinter. Affecting 10% of the global population and responsible for more than 1 million deaths each year, Chronic kidney disease has become a major public health issue. In the US, along 30 million people, or 15% of adults, are estimated to have a chronic kidney disease. Around the world, the number of people with chronic kidney disease continues to rise. We use Omentum as a bioink for 3D bioprinting. Omentum is a layer of peritoneum that surrounds the abdominal organs. Omentum contains many growth factors for renal regeneration. Moreover, it is known as a safe material because it has been used in clinic for more than 100 years. According to glomerular filter rate, CKD is divided into 5 stages, and we have a two treatment model. First is Omentum patch, which is treatment for patient with CKD stage 3 to 4. And for stage 5 CKD patient, we will use decellularization and recellularization kidney. For more detail, please follow next section technology.
Hi, this is Boyoung Choi from Lockheed Healthcare. Today, I am presenting a session on application of the Omentum patch to reduce renal fibrosis. Here is a schematic image that explains the whole process of our technology for kidney regeneration. First, autologous tissue is extracted, which is then prepared into bioink through micronization. Our patch is printed by 3D printer and then transplanted into the kidney. Unlike traditional treatments such as dialysis or kidney transplantation, our process is easy and simple, so it can be performed within 1 hour in the operating room. Our technology based on AI utilized in the patch printing. Lockheed's AI technology calculated kidney size and GFR to create a patch customized to the patient. Our team evaluated the antifibrotic effects of Omentum patch to reduce renal fibrosis induced by unilateral urethral obstruction in rat models. The patch is implanted in the capsule layer of the kidney as shown on the right video. As seen in the video, none of the bleeding issues happened during the transplantation. With the results of the transplantation, our published paper was chosen as the cover for the Journal of the Tissue Engineering Part C. I show that the result of the effects of Omentum patch on renal interstitial fibrosis induced by UUO in rats. From the heat map using RNA sequencing, fibrotic-related genes were upregulated in the fibrin patch group, as shown red color. But in the Omentum patch group, expression of these markers are lower rather than fibrin patch group. From the result of quantitative analysis of figure B, the expression of fibrosis markers such as ACTA2, CTGF, TGFB1 were significantly lower in the Omentum patch group compared with that in the fibrin patch group. In particular, HAVCR1 and LCN2 genes in several forms of CKD correlated with tubular interstitial fibrosis and tubular cell damage were greatly reduced in the Omentum patch group compared to fibrin patch group. We analyzed differentially expressed genes in order to identify the pathway associated with the genes from RNA sequencing data. All data show the momentum patch helps kidney recovery by regulating the balance of genes, abnormally up and down regulated by UUO. A geo-enrichment was conducted to confirm the cell mechanism related to gene of the momentum patch group. Of interest, many genes related to epithelial cell proliferation or migration were upregulated in the momentum patch group. In addition, the genes related to linear tubule development was upregulated in the momentum patch group, compared with that in the UUO control group. The reconstruction of linear tubule plays critical roles in kidney tissue regeneration. We perform empty staining and serous lead staining to evaluate interstitial fibrosis in kidney cortex after 2 weeks of UUO. On the right side of the bar chart, momentum patch group in UUO was significantly reduced. These findings suggest that the momentum patch plays a role in lowering linear fibrosis in the lead UUO model. Our group also confirmed safety of the momentum patch from Purkinje model. In this part, this study applied the momentum patch to the kidney of Purkinje models. We confirmed the safety of transplanting the momentum patch in 7 micropics for 12 weeks. In the Purkinje model, the same surgical procedure was performed as in the lead experiment. Momentum was harvested, preparing the bioink by micronization process. Print momentum patch and then transplant into capsule layer into the kidney. The safety of patch implantation was evaluated for 12 weeks after surgery. Biochemical profile analysis like albumin, BUN, and creatinine, the renal functional capacity showed no significant difference from normal ranges. In the H&E staining, we confirmed that after patch transplantation, the kidney retains the normal histological structure of linear tubules and glomeruli. We also check linear vascular resistance by calculating the RI value using ultrasound Doppler examination. Therefore, the data suggests that patch transplantation is safe for animals without causing any damage or complications. There are a lot of advantages of our technology based on the momentum patch results from the previous slide. As we apply process immediately after extracting the momentum without any enzyme, incubation and centrifugation, surgery time can be saved. Also, using autologous tissue minimizes the immune response. It can do one-time operation. It was also approved for minimally manipulated process from EMA. Based on the advantages shown on the screen, currently IRB is in progress with domestic hospitals. Here is our technology procedure video. We have two options for clinical application based on the momentum patch results from previous slides. One, using the 3D bioprinter. Another, using laparoscopic surgery. You can do surgery with the kit you want. In our group, we seek pioneers to join us and save patients suffering from kidney problems. Thanks.
Hello, I am Boyoung Kim, a researcher of Lockheed Healthcare. Today, I am going to present a session on decellularization and recellularization technology. We have developed a technology that overcomes the limitations of organ regeneration and accelerates the realization of organ regeneration through decell and recell technologies. This technology regenerates organ by injecting human cells into the ECM scaffold while maintaining the organ's ECM scaffolds. This technology can revive the tissue structure that 3D bioprinting or organoid technology cannot implement. A major advantage of T-cell organ is that no immune response occurs because only the ECM remains and the host cell and DNA are removed. When a patient's cells are transplanted into a T-cell organ, with this technology, customized organ regeneration is possible without immune rejection. So, Lockheed began research to utilize this technology to compensate for the lack of organ transplantation. Last graph showed the number of people waiting for kidney organ transplantation from 1995 to 2010. Next graph shows the number of deceased donor kidney discards in the US from 1996 to 2011. As you can see, there's a lot of people waiting for kidney transplantation. So, Lockheed Healthcare aims to save people who wait transplants by using our technology, which decell and recell discards organs. Lockheed Healthcare has developed equipment necessary for organ regeneration research to realize these technologies. Decell and recell of organs takes 3 days and up to 50 days respectively, and it requires a long labor force of researchers and a lot of equipment. To solve this problem, Lockheed's Dr. in vivo rich region was developed as an all-in-one system and our process was automated. This equipment has various built-in functions such as clean system, incubator, negative press, debubble, perfusion system, and controlled by software developed by Lockheed Healthcare. In addition, FDA registration and patents have been completed. With success in organ decellulization of small and large animals using the corresponding our equipment. It can be applied to various organs such as the stomach, pancreas, and the reproductive organ as long as it is an organ that can be perfusion regardless of organ. In the case of regeneration, cells were injected into the renal artery, and it was confirmed that the cells settle and adhere to the glomerulus. In addition, by inoculating renal epithelial cells into renal DECM, it was also confirmed that epithelial cells return to the corresponding location in the glomerular envelope. It plans to do its best to succeed in developing customized bio-artificial organs specialized for patients by implanting patients' own cells into DECM and resell them with Lockheed's technology. As an actual example, cartilage currently used in plastic surgery is decellulized and used as a transplant material. Lockheed Healthcare developed its own decellulization solution and succeeded in developing a solution that removes factors that cause immune response in bone tissue. We would like to provide samples for researchers interested in Lockheed Healthcare technology and solution and DECM. Bioink, cell culture dish, and artificial skin can be used as various research cases. So if you are interested, please contact us by email. Thank you for listening to my session.
Hello, my name is Haewon Kim from Rocket Genomics. Nice to meet you. Thank you very much to those who have been waiting for the webinar so far. It is an honor to introduce Rocket Genomics single-cell analysis technology through this webinar. From now on, let's start introducing the single-cell analysis technology of Rocket Genomics and its application to kidney regeneration research. Rocket Genomics is a company that possesses the tissue dissociation technology of various tissues derived from humans and various animals and plants, as well as the technology that can analyze the genome and transcriptome and proteome of single-cell unit. Based on these technologies, we are globally providing single-cell multi-omics analysis services and discovering various disease diagnosis biomarkers and treatment targets in collaboration with various researchers. In addition, Rocket Genomics is working to develop hyper-personalized organ regeneration technology through cooperation with Rocket Healthcare. Single-cell analysis is currently a technology required by most bio-related laboratories and companies worldwide. Every tissue in our body, even a cancerous mass, is made up of a wide variety of cells. However, the existing bulk analysis technology could not distinguish the characteristic of each of the various cells and only knew the average of characteristics. Single-cell analysis is a technique that overcomes these limitations and allows for much more precise and accurate cell status. Therefore, this technology is accelerating the personalized treatment and precision medicine that all healthcare companies and research institutes around the world are aiming for. And it will enable more efficient drug development and biomarker discovery. So one question arises here. Why, even though this technology is revolutionary, is it still not being used in many laboratories? The reason is single-cell analysis is difficult to start and very expensive and difficult to analyze properly because of the huge amount of data available. In particular, when compared to the existing bulk analysis, there is a very large difference from sample preparation method, so many failures are experienced at the beginning. Rocket Genomics is working to popularize single-cell analysis technologies by solving these problems. So, what kinds of competitiveness do we have to overcome this problem? First, Rocket Genomics overcame the difficulties of getting started through research consulting and tissue dissociation technology. So far, we have the experience and data of conducting single-cell analysis from a total of 10 species of animals and plants and 52 human tissues. This gave us technology to create more accurate and valuable data. Second, we were able to provide analysis services at half the price of our competitors through the technology that enables cost reduction. Lastly, we solved the difficulties of big data analysis through customized analysis services. As a result, Rocket Genomics is providing a more professional and cost-effective services to customers. So, the kidney is an organ with a very complex structure and function and is composed of various parenchyma and immune cells. And single-cell analysis provides a powerful approach to investigate the intrinsic complexity of kidney and understand the diverse cell types and states that exist during kidney development and homeostasis and disease. As you can see in this table, not only Rocket Healthcare and Rocket Genomics, but also many groups around the world are already conducting research on various cell changes that occur in the kidney, in the process of kidney development and kidney transplantation and various diseases and disease models by single-cell analysis. Through these studies, a single-cell-based kidney atlas map was created, and all of this data has been made up into an open database. So all researchers conducting kidney research are comparing their own data to derive more valuable results. In the near future, personalized, non-invasive monitoring and early diagnosis and disease monitoring and prognosis prediction and targeted treatment will be possible through single-cell analysis from patient biopsies or urine to treat complex and serious kidney disease such as CKD or AKI. So Rocket Genomics together with Rocket Healthcare will do its best to treat numerous kidney disease patients around the world. If you are interested in single-cell analysis as well as kidney research, please feel free to contact Rocket Genomics. Thank you for your attention.
Have you enjoyed the time? Let's move on Q&A session. Before we start, we answer the FAQ. The first question is, what is the advantage of momentum patch? The kidney transplant patient need to take an immunosuppressive drug for their whole life. However, since the momentum patch is made of the patient autologous tissue, there is no side effect and immune response. So we have advantage. The patient doesn't need to take the immunosuppressive drug. Then next question is, when can these technologies be applied to patients? The decellularization and recellularization technology is still on the process, the research stage. However, the Momentum patch is now preparing the clinical trial. Please contact us more detailed schedules. The last FAQ is, how can decellularization equipment be automated with various functions? Let's look at the slide. Developed with a focus on the convenience of researchers, Dr. In Vivo Niche Region has an automatic system of the entire process using many sensors including water level, temperature, and negative pressure. A fully automated program system allows the device to supply and discharge solution based on present protocol at each stage. In addition, this process can be monitored in real time through the built-in cameras. A lot of questions are live. Please understand we only have a selected question because of time limitation. The first question is, what kind of platform does Rocket Genomics use? This question will be answered by Dr. Kim.
Yes, thank you. Now, Rocket Genomics is using two kinds of platforms. One is 10x Chromium, the other is BD Rhapsody platform. We think two kinds of platform have a best performance for single-cell RNA sequencing. In addition, we have a technology for single-cell RNA sequencing for gene expression, and single-cell ATAC sequencing, and single-cell multiome, and single-cell TCR and BCR sequencing, and single-cell targeted DNA sequencing. And in the future, we are going to develop many kinds of single-cell sequencing for multi-omics analysis. Thank you.
Yeah. And next question is, what are the advantages of micronized momentum? The native momentum is composed of adipocyte and milky spot. However, since milky spots are randomly distributed in momentum, so the active ingredient may appear uneven. We process our component evenly and make a patch. So therefore, the patch can supply the active ingredient to kidney very even state. The next question. Have you ever experienced kidney tissue through single-cell RNA sequencing? Please
yes.
answer the question.
Yes. Yeah, we have experienced the tissue dissociation of kidney tissue was, I think, it's really difficult. So there are so many heterogeneous cell types and debris, but we set up the protocol and success to make a single-cell RNA sequencing data. So this kind of experiments will be developed for collaborate research of Rocket Genomics and Rocket Healthcare. And we are analyzing the single-cell RNA sequencing data using the bioinformatics, many kinds of bioinformatics tools with our collaborators. So yeah, we have experience. And we're also looking for your project. So feel free to contact us. Thank you.
Next question, please.
Have you ever done multiplexing in Rocket Genomics? How was the result? Yes, it is really important questions. Yes, the multiplexing is also really difficult because all samples have have different cell viability and debris, many aggregates and cell numbers. However, we have really many experience for single cell multiplexing for many kinds of tissue types. So normally we pulled 6 samples for multiplexing, and about 40,000 of cells can be acquired. So it is not easy for single cell RNA sequencing for, using multiplexing, but we can now we can do well. So yes, we have many experience. Thank you.
And I will take a last question. Please understand, because of time limitation, we only select the question to answer it. The final question is, what is the difference between kit 1 and kit 2? That's a good point. There is no difference in the component of patch, but there is a difference, the way to plant the patch. Please look at the slide. The kit 1 is used to print out patches to a 3D bioprinter and transplant patch through a plank incision. Kit 2 is used to inject patches using laparoscopy. With kit 1, a wider and larger amount of patches can be transplanted into the kidney. Doctor can choose kit as a convenience method for their own treatment. We are recruiting pioneers to apply this technology to clinical practice. I look forward to hearing from doctors who are interested. Thank you for watching the webinar. We plan to present this data at World Congress of Nephrology in March. Please visit our booth, number 22. For other detailed inquiry, please contact us. Thank you.
Thank you.